To be or not to be? That is the question. Although I'm pretty sure Hamlet wasn't talking about cellular biology, this is the exact decision cells are faced with when a cancer patient undergoes treatment. The largest class of chemotherapeutics are called DNA-damaging agents. We used to think they worked by damaging DNA so severely that cells could no longer survive. What we now know is that it's not the damage itself that kills the cancer, but rather the cell that recognizes the DNA damage and makes the decision to activate autonomous suicide pathways. Hamlet unknowingly articulated this decision for cells to either passively suffer the slings and arrows of outrageous fortune from chemotherapy, or to take action and kill themselves, the result of which can literally mean the difference between life and death for a patient. Over 500,000 Americans died from cancer last year, showing us that clinical failures such as drug resistance and cancer reoccurrence are still challenges faced by many. So why do these drugs not always cure the cancer? What is happening at the molecular level to tip the balance of whether or not a cell chooses to initiate its self-destruction? The answers to these questions will provide us with ways to more consistently eliminate this disease. By targeting the link between the cell recognizing the DNA damage and initiating these pathways, we can delete this decision-making process altogether. And in doing so, give the cancer no choice but to destroy itself. So you see Hamlet on my slide, but instead of staring at Yorick's skull, he's staring at damaged DNA. Now, my thesis work focuses on the cellular response to the specific type of DNA damage pictured in his hand, called DNA protein crosslinks, or DPCs. Now, DPCs are formed by these chemotherapeutics when a protein becomes permanently linked to the DNA of a cell. Previously, the cellular response has been really difficult to understand because the damage was being created randomly throughout the genome. My strategy is to create a site-specific DPC on a chromosome and track it in cells. Using this method, I can test specific hypotheses regarding how the size and location of the DPC sways the cell's decision to initiate these suicide pathways. Once understood, these pathways can then be selectively targeted, and in doing so, pharmacologically force the hand of the cancer cells to take arms against a sea of troubles, and by opposing, end them. Thank you.